Have you ever wondered what would happen if you took human DNA and put it into a mouse? This might sound like the plot to a science fiction movie, but for me, it started with a cartoon. As a child, one of my favorite cartoons was Pinky and the Brain, a show where researchers took human DNA and put it into mice, and the mice became smart enough to start devising plans to take over the world. This, to 9-year-old me, was very funny. But to adult researcher me, who will be 44 in just 9 years, this was actually intriguing. In my lab, we study cerebral cortex development, and the cerebral cortex is the outermost part of the brain responsible for many of our higher level processes, such as reasoning and consciousness. And I study this in the mouse, and the reason why I do this is because on the surface, these two brains look very different, but much of the cell biology is conserved. For example, both have neural stem cells that are capable of going on to make the two principal types of cells in the brain, known as neurons, which conduct electrical impulses that most people recognize as brain cells, and then another class of cells known as glia. And the glia cells support the neurons by helping with things like energy production and removing waste. And you can imagine a bigger brain needs much more of these glia to keep up with the output. And so that's one difference between the human and the mouse. Another difference is in the cortex. You might have heard that humans have a larger cortex than you would expect based on body size, which is true, but it doesn't tell the whole story. And to tell the whole story, you need to divide the cortex into upper layers and lower layers. And if you were to do this and divide the lower layers by body size, there's no difference between mice and humans. However, if you looked at the upper layers, there's this drastic increase in upper layers of the cortex, which is important because this contains cells we call upper layer neurons, which are responsible for many of the functions we think about, such as reasoning and consciousness. And so I wanted to know what would happen if you took human DNA and you put it into a mouse. How would the brain be different? And so I did this experiment, and what you see is that there's a drastic difference between the control condition and the condition that has our human-specific sequence. But on the cellular level, it's much more exciting, because at the cellular level, we see that the human DNA sequence containing brains actually have more of these specialized upper layer neurons and more glia, which is exciting not because we created talking mice, but because we understood more about how the human brain evolved. And this is important in things like neurological disorders and how they arise. And so by taking this DNA sequence and understanding cell by cell, we start to understand how the human brain is capable of doing things like creating cartoons about mice that try to take over the world. Thank you.